Hi, everyone. Good morning. Welcome to today's webinar from the Hepatitis B Foundation and Hepatitis Delta Connect. Thank you so much for joining us this morning for our presentation from Dr. Ira Jacobson, who is a professor and professor of medicine and director of hepatology at New York University's Langhone Medical Center. And he'll be speaking with us today about hepatitis delta virus, giving an overview of the virus itself and latest treatments. My name is Beatrice, and I work for the Hepatitis B Foundation and manage the Hepatitis Delta Connect program. And we're so excited for you all to join us today. So just some housekeeping notes at first. So everyone is going to be on mute throughout the course of the presentation, and we will have time for questions at the end. So if you do have any questions, please feel free to put them in the chat box and we will get to them at the end of the presentation. This call is going to be recorded, the full webinar, and it will be emailed to all participants along with the evaluation form at the end. So if you missed anything, you can always go back and view the recording. We do have some surveys that we would love it if you could fill out that are specifically designed for both people living with hepatitis delta and providers, so that if you'd like to get contacted by us for not very frequently, but just sometimes for to be added to our newsletter or to learn updates about clinical trials, et cetera, please feel free to fill out the surveys and join those lists, which will be posted in the chat box. And then just a quick overview of the program. We will start with an introduction of Dr. Jacobson by Dr. Tim Block, who is the president and co-founder of the Hepatitis B Foundation. And then we will go into Dr. Jacobson's presentation and then have a short story from a storyteller who's living with hepatitis delta, and then have time for questions and answers at the end. So thank you all so much again for joining us. And we will begin with an introduction of Dr. Jacobson. So Dr. Block, you can feel free to take it away.
Thank you very much, Beatrice. So as Beatrice said, I'm Tim Block, president and co-founder of the Hepatitis B Foundation, but we're in for a real treat today and a lot of information because of the seminar from Dr. Ira Jacobson. And again, if you have questions, please put them in the chat box. And I know Dr. Jacobson will get to them as he can throughout the talk or at the end of the talk. So as Beatrice said, Dr. Jacobson, yes, he's director of hepatology at NYU Langhone Health and NYU Medical School. But what he really is one of the world's thought leaders in the area of viral hepatitis. He trained at Columbia, UCSF, and the Harvard Medical School systems. So he's got that pedigree, of course. He has published more than 250 papers in the field. And I want to say some of these papers are the definitive papers in the area of treatment of viral hepatitis, cited thousands of times, placing him in terms of his impact factor and contributions in the upper 1% of all scientists and clinicians in the field today. He's received numerous awards. He was the American Liver Foundation Physician of the Year in 2007. But maybe most interestingly, he's also now a fellow of the Royal College of Physicians from England and London. So he, maybe most importantly to us today, he has led some and participated in the definitive clinical trials and evaluations of medicines for viral hepatitis, hepatitis B, hepatitis C, and of course now hepatitis delta. So it's a real privilege to be able to hear him and be able to host him from the Hepatitis B Foundation. So without further ado, because I know we've gone on, I have actually several pages of his accomplishments. Let's just get right to you, Dr. Jacobson. Thank you very much, and I will go to mute.
Thank you, Tim. It's a great honor to be here with all of you, and to get an introduction like that from you makes this day a very special one for me. I haven't visited you in a while. I think I'm overdue. So I'm hereby inviting myself at your earliest convenience to revisit your institute and the wonderful work that you've done. I've also always enjoyed learning from you. You've been one of the clearest explicators of hepatitis biology, treatment, and all its other aspects that I've ever known. So it is a privilege to talk to you today about a major worldwide public health problem that, if anything, probably hasn't gotten enough attention over the years, but I think is getting its fair share now, and that is hepatitis B virus. You can go on, please. Very quickly, these are my disclosures coming up. I have done a little bit of work with MIR, which is supporting the program. And next, please. So hepatitis B is really a unique virus, at least in the animal kingdom. It's similar to things called satellite viroids in plants. It's been estimated that about 15 to 20 million infected people exist or living around the world. 60 million is the highest estimate that I came across in my review of the literature that I conducted for this program. One recent major paper that I'll show you estimates it at 40 million. So it's somewhere in this range, probably in the middle. About 5 to 10% of people around the world who have chronic hepatitis B are infected with hepatitis D. Hepatitis D is uniquely dependent on the coexistence of hepatitis B because it is a defective virus in the sense that its genetic code cannot account for the production of its own coat protein. All viruses have envelope or surface or coat proteins, kind of like our skin, if you will. And hepatitis D is so simple in its genetic makeup that it can't even code for its own coat protein. It relies on the coexistence and nearby presence in its neighborhood in liver cells with hepatitis B virus making its coat protein for it. Indeed, it only codes in its genetic code for only one protein called the hepatitis B antigen, which exists in both a large and a small form. Hepatitis D can only infect people with hepatitis B, but it can do that in two ways. It can co-infect people acutely or suddenly who have never had either of the viruses before, or more ominously, because of the much higher rate of chronic infection and the consequences that can ensue, it can superinfect chronic hepatitis B carriers even after many years of pre-existing infection with hepatitis B alone. Most patients with chronic hepatitis D are negative for an important protein of hepatitis B virus called E antigen, and this has to do in general with a lower replicative state and either inactive carriership from the viewpoint of hepatitis B perhaps no longer replicating, or the presence of certain mutations that make the hepatitis B virus unable to produce its E antigen anymore, but still can replicate and cause significant chronic liver disease. I won't go into the technical nuances of the E antigen system any further today unless there are questions. The main problem with hepatitis B, it's quite pathogenic. It confers a high risk on affected patients of progressive scarring with fibrosis, cirrhosis, which means extensive fibrosis, and the ever-present risk in patients, particularly with advanced scarring of the liver, of liver cancer, which we call hepatocellular carcinoma or HCC. For many years, interferon has been the only treatment. This is the drug we've gotten away from in hepatitis C and to a large extent in hepatitis B, though there is some usage of it. But until now, interferon has been the only drug that we really have had available, and it has significant limitations on its effectiveness and the requirements for rather prolonged therapy. One of the most exciting developments that I'll focus on in the second half of this talk is the promise offered by new drugs on the horizon. Next slide, please. Again, this is this really very simple virus, which has a single-stranded RNA genome. Some viruses have DNA genomes, just like we do. But in this virus, like many others called RNA viruses, RNA comprises the genetic code, if you will, of the virus. And as mentioned before, it codes for a protein called the delta antigen or hepatitis B antigen. Viral proteins are synonymous with antigen for the purposes of this talk. And then that coat protein that the virus cannot produce on its own, called hepatitis B surface antigen. On the right is a picture of viral particles under an electron microscope. Next slide. So I always enjoy history and biography in my life in general, and I really enjoy talking about the history of science and great discoveries in science. And it was an Italian investigator who was working at the National Institutes of Health in the late 1970s, a young investigator at the time named Mario Rossetto, who's just as nice a person as he appears to be in this picture. who discovered hepatitis B, and I'll just give you a brief glimpse of some of his seminal papers. Next slide. The first one, and perhaps the most famous because it started all of it, was when he called a protein that he had discovered an antigen by studies called immunofluorescence studies in the livers of people infected with hepatitis B, discovered a new antigen that he called Delta, which kind of conferred an element of an aura of mystery to it, I think. And he quickly determined that there was a corresponding antibody and that this antigen antibody system was associated with hepatitis B virus in the liver and in the blood of carriers of hepatitis B surface antigen, which had been discovered over a decade before. The person who discovered the hepatitis B surface antigen was a gentleman named Baruch Blumberg, who Tim knows or knew very, very well. He's unfortunately not with us anymore. Dr. Blumberg won the Nobel Prize for discovering this protein, which he first called Australia antigen, because that's where he discovered it. And he made many discoveries after that that ultimately linked that mysterious protein to hepatitis B. Many speculated after Dr. Rossetto discovered this virus that came to be called hepatitis B, or the Delta agent, as I'll show you in a moment, that he too might win a Nobel Prize one day. We're happy that our colleagues in hepatitis C, including my dear friend, Professor Charles Rice at Rockefeller, won the Nobel Prize for hepatitis C, along with his two co-winners. We're hoping that maybe one day Dr. Rossetto will receive the international recognition from the lay public that he deserved in some additional form or another, but he's a great figure in the history of viral hepatitis. Next slide. The next paper was on the incidence and significance of antibodies to this new antigen called Delta in HBV infection. Next slide. And then for the first time in this paper, in the Proceedings of the National Academy of Sciences, he no longer just called it the Delta antigen. He and his colleagues who were very famous investigators like Dr. Purcell and Dr. Jaron at the NIH had figured out that this was actually a new virus that seemed to coexist in the livers of people with chronic hepatitis B. And they were able to transmit the virus in the day when you could still do chimpanzee studies to chimpanzees, and they were able to describe the clinical and serologic events that ensued after such infection occurred. Next slide. And then finally, he began to write what became a long series of papers and ultimately joined by many other investigators on the epidemiology of this agent, which I'll talk about in a little bit. So these are just four of his initial papers that really created the standard in this field. Next slide. This is one map of the global distribution of hepatitis D infection among hepatitis B surface antigen carriers. And on this slide, red is bad in the sense that it's associated with a very high prevalence of hepatitis D infection, over 50% in the darker red color that you see in Mongolia, parts of the Amazon basin, parts of Africa, and the Middle East, and so on, areas of intermediate prevalence, and then areas of relatively low prevalence of hepatitis D in B infected people, shown in green. Next slide. That's from 2019. The next slide shows that even amongst people of epidemiologic bent who've done extensive studies on these things, there's not total agreement. It depends on what databases you use and how you analyze them. Here's a slide that shows only Mongolia with a prevalence of hepatitis D of over 50%, with the other areas shown in red previously being a bit lower. But the point remains the same, that these areas shown in various colors are the areas that we would consider endemic, if you will, for hepatitis D. Next slide. Now, this was a recent meta-analysis of these prevalence papers in the global population that was published in a leading hepatology journal just a year ago. This is a journal of the American Liver Association. And you can see that they looked at 182 articles from 61 countries and found that the overall prevalence around the world of hepatitis D was about 1 in 100 people. But the pooled prevalence in B-infected people was upward of 14 or 15%. And the seroprevalence, which means the presence of antibody, was about 11% in a mixed population without particular risk factors for hepatitis B, like IV drug use and high-risk sexual behavior. It was 38% in the IV drug-using population and 17% in the high-risk sexual behavior population. Next slide. These are areas listed in another very recent paper that just came out this month. as endemic, and these, I'll give you just a second to take a look at it, correspond to the countries that were shown in various colors other than pale gray or other more dull colors, denoting low prevalence in the previous two slides around the world. These are the areas in green or red. Next slide. This gets back to the paper just published this month. And this is from the prestigious NIH group, which continues to be prodigious in its accomplishments in the field of viral hepatitis. This reflects essentially all the patients who've been referred to the NIH for one reason or another for hepatitis B. 3,000 people were tested for hepatitis B, of which 652 were positive. They managed to test almost all of their patients, 90% for hepatitis B antibody, which, as you'll see again, is the main marker that we use to screen for hepatitis B infection. Although, as you'll also see, it doesn't always correlate with the actual presence of virus. Nearly 20% were D antibody positive. Now, that doesn't necessarily reflect the true prevalence in the United States or in North America, which is the cohort that's represented in the slide. Because there's a referral bias. If you're referred to the NIH, there might be a particular reason that somebody referred you, whether for experimental treatment or unusual features of the case or more severity, greater severity of the case, and so on and so forth. 80% were confirmed to have chronic hepatitis B infection by HBV or hepatitis B RNA testing. It's a PCR, and we all know about PCR these days for all kinds of reasons, but when you have a nasal swab for COVID, for example, that's a PCR. It's a powerful technique for which its discoverer won the Nobel Prize. Again, mentioning the Nobel Prize a number of years ago, because it's been an intrinsic development in medicine, forensics, and many other fields. Now, among those with hepatitis D antibody, just to mention the E antigen system again, only 10% were E positive. And I guess I will say one more thing about the E system, which is that when we say a hepatitis B patient is E positive, that generally is associated with a particularly high level of viral replication. And the point here is that 80% of the patients at the NIH had the E antibody, the E antigen had cleared. They had E antibody with lower or sometimes undetectable levels of hepatitis B viral DNA, which is how we measure viral loads in hepatitis B. And the point that I'd like you to take away is that active hepatitis B infection usually is associated with lower levels of replication of hepatitis B. I'm not enough of a scientist to give you the precise mechanisms, but you can almost think of it as hepatitis D and hepatitis B competing for the same kind of ecological niche or space within the cell, perhaps for resources that the viruses need to produce themselves. 65% of the patients of the NIH were from an endemic country, 11% were IV drug users by history, and 12% were on nucleoside treatment or nucleotide treatment for hepatitis B. Notably, hepatitis D antibody positive or exposed people were more likely to be from an endemic country or be IV drug users compared to so-called unexposed patients. Next slide, please. The authors, because of these associations they made with various risk factors, recommended screening for hepatitis B with any of the following: history of IV drug use, a low level of hepatitis B replication for the reasons that I mentioned before, evidence of active liver irritation or inflammation. And for those of you who aren't completely familiar with it, the ALT is an enzyme in the liver cells that performs certain basic chemical functions there that leaks out of liver cells if they're irritated. Indeed, most of the patients who come to my hepatology practice for consultation for any reason come because a physician found that they had a high ALT on a routine blood panel, and now it's left up to us to figure out what's causing the liver injury. So if there's evidence of active liver injury, that makes hepatitis B a little more likely because of the pathogenicity or tendency to cause liver injury that hepatitis B virus is associated with. And finally, origins from endemic country. Now, many clinicians, including myself, and I see my dear friend, Dr. Gish, the medical director of the Hepatitis B Foundation on the line, have an alternative view, and I hope I'm speaking accurately for Bob, but I think so. We believe that at least on a one-time basis, all persons with hepatitis B manifested by the presence of hepatitis B surface antigen in the blood, which is the cardinal feature of chronic hepatitis B infection, should have a one-time test for hepatitis B. I do have to say that my yield, having done that in many hundreds of patients in New York throughout my now 36-year career here in New York City, the yield has been very low. I think Dr. Gish would tell you that in his area, the yield is perhaps somewhat higher, and he's actually published on that. But I never want to miss a patient with hepatitis B because it has such profound implications for management. So I screen everybody. Next slide. So this shows the basic structural features of hepatitis B and hepatitis D. And of course, there's really no time to go into this extensively, but remember you have that hepatitis B surface antigen, which is the coat protein or outer envelope protein of the virus. And then there's an inner protein called the core antigen, which is shown on the lower left. Can you all see my pointer if I use it? Beatrice, can you tell me? Do you see it? If you don't see it, I won't use it.
Yeah, I don't think so.
Yeah, that's fine.
Okay.
I'll do fine without it.
Okay.
And then inside the viral particle for B is that double-stranded or mostly double-stranded DNA genome, the genetic code of the virus, as I like to put it. Hepatitis D is a bit smaller as a viral particle, and again, quite a bit simpler. It has within it the single-stranded RNA of the virus, a completely different genome, genetic makeup, than the DNA genome of hepatitis B, and again, lacking the ability to make its own coat protein. At the bottom on the caption, we see that delta antigen or hepatitis B antigen, the protein coded for by that genetic code of the virus, can exist either in a small form or a slightly larger form. So we have the small and large delta antigens. And that little yellow elliptical or oval shape represents farnesylation, which is just the addition of a small lipid or fatty particle molecule to the protein in a process called prenylation. And the reason I'm bringing that up is I'll talk about it again later when we talk about one of the newer treatments for hepatitis B. But the integrity of the viral protein in the case of hepatitis D depends on that farnesylation process. So you can already jump to the guess that if we can inhibit that somehow, it might be a way to inhibit HDV replication. And that's what's happening with a drug called lonafarnib right now. Next slide. And this is the life cycle. I'm not going to go into it in detail, but I will show you on the top of this slide a viral particle that's just aching to get into a liver cell and infect it. And in 2012, there was a great discovery actually out of China, where after decades of looking for it, it was discovered that the main receptor, the gateway by which this hepatitis B enters liver cells, is through a bile acid receptor that's abbreviated NTCP, and this has facilitated an enormous amount of additional research, and now the development of the first approved drug, at least in Europe, for hepatitis D, an entry inhibitor that we will talk quite a bit about in a little while. So needless to say, if this is the way hepatitis B gets in, it's the only way hepatitis D can get in, because hepatitis D has to be packaged in a coat of hepatitis B surface antigen or protein. Inside the cell, the virus gets into the nucleus where it reproduces, its proteins get produced, eventually new viral particles are packaged and secreted from the cell. And the three highlighted areas in red here show potential opportunities to intervene therapeutically or treatment-wise in people with hepatitis D. First, the concept of inhibiting the very entry of the viral particle into the cell, which we call entry inhibition that I've talked about. Then things called nucleic acid polymers, which I'll talk about toward the end of the talk. And finally, the concept that I just mentioned on the previous couple of slides called prenylation inhibition by that drug lonafarnib. Next slide. There are different hepatitis B viral genotypes, and genotype 1 is the most prevalent worldwide out of the 8 genotypes, predominant in Europe and North America. I'll let the rest of the slide speak for itself in the interest of time. This genotype testing is not approved in the US and is rarely done or accessed by clinicians. Next slide. This is the global distribution of hepatitis D virus genotypes. And again, I'll show you the preponderance in many parts of the world, including here in the US, of genotype 1. But if you see the red areas in South America, particularly the Amazon basin, that is genotype 3. And genotype 3, at least in people in South America, reminds us of genotype 3 by sheer coincidence in hepatitis C, where it took us years to figure out that genotype 3 was the genotype of hepatitis C most likely to cause cirrhosis or scarring of the liver. That's what happens to people infected with genotype 3, at least in the South American region. It is considered the most pathogenic form of the virus, followed by genotype 1, which seems to have greater pathogenicity than genotypes 2 or 5. Next slide shows some of this. Diagnostic testing for hepatitis D is mostly, as shown by the arrows on the left, through antibody testing and by doing PCR for hepatitis D RNA. There is, on the second line here, a delta antigen test. Remember, that's the protein that Dr. Rossetto initially discovered when he was doing research on liver tissue from people with hepatitis B. And there is such a test, but it's not used very much clinically. And you'll see my algorithm for what tests we ought to do at various stages in the evaluation for hepatitis D shortly. Next slide. So this is what happens when you get hepatitis B and D together. The serologic course is followed by the appearance of the hepatitis D viral RNA, as well as the new or de novo appearance of hepatitis B surface antigen. If you've been infected with both viruses for the first time, these are the two markers that initially appear. This slide shows the antibody response, and there are different types of antibodies in our bodies called immunoglobulins or Igs. An IgM antibody characterizes acute infection from most viruses. So if somebody comes into our office acutely ill with a suggestion from their history that they've acquired an infection recently, an IgM antibody is the one we use for those viral infections, including hepatitis D. This is eventually replaced by IgG, which is related to more time after exposure. But in people who get hepatitis B and D together, they have the same chance of clearing hepatitis B infection, say in immune-competent adults of 95+%, that other people do. The hepatitis D does not seem to make hepatitis B more likely to become chronic. So when the hepatitis B resolves, the hepatitis D resolves with it by definition, because the hepatitis D can't do anything if you don't have hepatitis B anymore. There are some nuances to all this that I won't go into, and my colleagues will recognize what I'm talking about, but you'll see here that this happily after ending, or no, ending happily ever after kind of resolution of co-infection is marked by the long-term persistence of antibody to both hepatitis D and hepatitis B in the form of surface protein antibody as well. Next slide. This is really where the bigger problem is. This is people who get hepatitis D in the setting of already existing hepatitis B. And this is an easy thing for hepatitis D to do because the machinery in the person's liver for making lots of hepatitis B surface antigen or protein is already running at full factory capacity. So hepatitis D gets into a liver like that, and it's very happy because the surface protein is already being produced in abundance by the hepatitis B infection. And now the hepatitis D can plant itself very firmly and quickly in the liver and start to chug away. And these patients don't clear their hepatitis B. It persists indefinitely in many, if not most, shown in purple here by the long-term persistence, not only of the hepatitis B surface antigen, attendant upon chronic hepatitis B infection, but the long-term persistence of the hepatitis D RNA. And the red line shows that long-term inflammation of the liver generally ensues with chronic hepatitis B infection as well. These patients have high levels of total antibody to hepatitis D. Most of it is the type called IgG. And this is why anti-HDV or hepatitis B antibody is the usual test that we use to screen people with hepatitis B for hepatitis B. Next slide. The algorithm for the evaluation of hepatitis B, taken from a recent review article, follows from everything I've just said. We have a hepatitis B surface antigen positive patient who we want to screen. In the second line, we get a hepatitis D antibody. We don't get a delta antigen that is less commonly present, less reliably present in people with chronic hepatitis D. We get an antibody. And then if the patient's positive, we need to check the hepatitis D RNA, whereupon we evaluate the state of their liver disease and ultimately consider therapy as shown on the lower right. But the real point on this slide was to show that the initial step in a B-infected person is to get an antibody to D and then an RNA to confirm whether the infection is actually still present and ongoing. Next slide. This slide shows the natural history of hepatitis D infection. Remember that if a normal liver is infected with hepatitis B, at least in adults, that chronic hepatitis B ensues in less than 10%, really around 5% of all such infected people. But the eventual outcome can be the development of liver scarring or fibrosis. And a patient on that pathway can easily get infected, as we've said, with hepatitis D for the reasons that I've mentioned, which gives them a higher chance, unfortunately, of developing cirrhosis and liver failure and/or hepatocellular carcinoma. Next slide. This is an estimate from a recent review of the adverse outcomes associated with hepatitis D. Cirrhosis is 2 to 3 times more likely to ensue in a patient who has both hepatitis D and hepatitis B, unfortunately, compared to hepatitis B alone. Liver cancer is at a relatively high risk. The need for liver transplantation, liver decompensation, which is failure of the liver to maintain its normal functions, is increased in risk, as is unfortunately mortality. And this really shows why it's so very important to be developing new drugs for hepatitis D and prevent these terrible complications. Next slide. This shows the long-term clinical outcomes in patients with chronic hepatitis D. These authors looked at a couple of thousand hepatitis B infected subjects, of whom 5% are positive for hepatitis D antibody. They looked at 118 of them with RNA. And the reason I'm showing this is to emphasize that about 3/4 of the antibody positive people, that is with Delta or D antibody, end up having RNA. Now, when I was young and writing my first papers on hepatitis D, we thought that if you had hepatitis D in the setting of ongoing hepatitis B, that you were stuck with ongoing active infection for good. But this slide shows what's come to be accepted, which is that a certain percentage of patients do have spontaneous subsidence of hepatitis D throughout the patient. Also shown in this study is a fairly high prevalence of cirrhosis, mostly in people with ongoing active hepatitis D infection, which makes sense, as manifested by RNA. When we've screened people for clinical trials, and we initially thought we had a patient for a trial because they were positive for D in the setting of chronic B, sometimes we've been disappointed to find that they no longer have the hepatitis D RNA, and therefore aren't eligible for a trial that is intended to treat people with active infection. Next slide. This shows the same point. I think I'll just go on. And this shows again that long-term clinical outcomes in patients with chronic hepatitis D are associated with whether or not persistent viremia is present. Next slide. These are new data from a paper that just came out in the main European Journal of Liver Disease, called the Journal of Hepatology, where they looked at a large number of D positive patients by antibody and showed that 88%, higher than the 73%. So there is some variability there, were in fact RNA positive. Most were immigrants from the various countries. You can see the genotype breakdown. These are all in France, by the way. A high prevalence of cirrhosis, 28%, half of whom had at least one episode of failure of the liver to function in one way or another with a clinical complication that qualifies you for the term of liver hepatic decompensation. At the end of follow-up, half the patients had cirrhosis, showing the pathogenicity. That's after a median of only 3 years. And naturally, persistent replication of virus was associated with more adverse outcomes. Next slide, please. Now I'll get to the treatment part of the talk, and this is a nice summary in a recent table by Drs. Zhang and Urban. Some of you will recognize Dr. Urban's name. He is an internationally renowned hepatitis B investigator who actually developed Mircludex, or now called Hepcludex, which we'll talk about. But the main point here is that these are the drugs that I'm going to talk about. The only drug that is still available in many areas of the world is interferon. So I'll show you a little bit about the history of interferon for hepatitis B. Next slide. And the current algorithm for the current management of hepatitis B is shown here, featuring interferon from, again, a recent review article showing that for active hepatitis D, the quote standard treatment has been pegylated interferon, mostly in the form we call alpha-2A, which is self-injected once a week for a year. It can be tough going, but we've used it for many years for hepatitis C, although it's no longer used there at all. Still favored by some for hepatitis B, and actually considered one of the frontline drugs for hepatitis B, along with the more commonly used oral agents. And the point here is that this slide shows you how we go about treating patients, what we look for. I think because of time considerations, I'll just also emphasize the point at the bottom, which is that clinical trials with novel therapies are a very important option to be able to offer patients nowadays. particularly in geographic areas where such trials are ongoing. Next slide. These are the clinical trials for PEG alpha interferon from 2006 to 2011. And I draw your attention very quickly to the rightmost side of the slide, showing sustained virologic response, which is negative PCR for hepatitis D RNA, a period of time, usually 24 weeks after the end of a course of treatment, which in these studies was either for a year or a year and a half, ranging from 17 to 43%, but mostly centering around the most off-quoted figure of about 25%. Next slide. Now, this is the HIT-1 study, which is one of the two major studies that have defined the role of interferon for this disease. And this study looked at the virologic response to treatment as determined again by serum levels of HBV RNA. There were three treatment groups, as you can see, PEG interferon and an older antiviral drug against hepatitis B called Adefovir. Two treatment groups. There was PEG interferon alone, 3 treatment groups, and Adefovir alone. And it was 48 weeks of treatment, 24 weeks of follow-up. And the most important set of numbers that I want to show you are the numbers in the first graph on the left. Second part of that graph is week 72, end of follow-up. So a year of treatment, 24 weeks of monitoring, and you'll see that 26% of the patients who got interferon plus Adefovir, 31% who got PEG interferon alone were negative for HBV RNA, consistent with that figure of 25% from an overall analysis of the studies that I showed you before. The Adefovir didn't seem to add anything to the PEG interferon. As you can see, these numbers were statistically not significantly different. Next slide. The HIT-2 study is the second more recent study on PEG interferon. This one was done with a newer antiviral for hepatitis B called tenofovir that some of you may be familiar with versus placebo. So it was interferon with tenofovir or interferon with placebo, 2 arms now for HBV infection, given now for 96 weeks, and even taller order to take interferon for 2 years with its side effects, but most of the patients managed. And I draw your attention to the boxed portion at the upper right of the slide, particularly the week 120 or SVR, sustained virologic response figures, showing how many patients were HBV RNA negative at the end of the 24-week follow-up period. And these numbers should seem very familiar to you. They're almost the same as the previous study, 31% and 23%. Again, when you look at the statistics, they were not significantly different. So the extra year of the interferon, at least in this study, didn't seem to make much of a difference in terms of inducing a sustained virologic response. Next slide. And I'm going to skip through this to the slide following the next one. This is a recent study just presented at the American Liver Association meetings several months ago of 13 patients, showing really something that conceptually I wanted to get across, which is this was a study of patients who've been treated not in a particularly systematic way like the rigid criteria for a protocol would dictate, but just kind of in clinical practice with a mean treatment duration of several years, as you can see. Sometimes the treatment courses were not consecutive. There may have been an interruption, patients put back on treatment, but the point of the study, as shown in that kind of stepwise picture on the lower right, is that the more you treated patients over the years, the more of them had a cumulative virologic response. And some of them even lost hepatitis B surface antigen, which I would call, if you will, the holy grail of hepatitis B therapy, to lose that last serum marker of hepatitis B infection. Next slide. This is an algorithm. We've already done this. Next slide. And now to the hepatitis treatments in development, one of which has been triumphantly approved recently to our delight, Mircludex, at least in Europe. This is a picture courtesy of my friend, Dr. Gish. He kindly sent me the other day, showing again that viral particle just aching to get into a liver cell, attaching on the left side to the bile acid receptor called NTCP that I talked to you about. The virus gets into the cell, it's uncoded, its genetic material gets into the nucleus of the cell, it starts replicating. I mentioned that prenylation process by which the large hepatitis D protein, which is about 28 or so amino acids larger than the small protein. They're almost the same, but not quite. One is a longer chain than the other. Remember that proteins are like chains with links. They're analogous to amino acids, the building blocks. So the larger protein is a slightly longer chain with a few more links than the smaller protein, but they're both needed. And again, you have to prenylate the larger protein to have an intact kind of functional viral particle. And then, of course, the virus that's produced has to get out of the cell. There's been a lot of interest in blocking the release of hepatitis B surface antigen or anything that's coded by hepatitis B surface antigen recently, as I'll show you. Finally, the green insert on the upper right shows you that we haven't forgotten about the possibility of antiviral activity as well as immune boosting from interferon. And there's a type of interferon called Lambda Interferon that's not in widespread practice, but has been investigated recently. Next slide. Next slide. So this is Mercludex B, which Professor Urban, shown on the right. I got this from Google Images, and there's a caption underneath, European Commission approves Mercludex B, which was a major development in August of 2020. And that's a smile on his face that I would infer was related to the announcement, but he's a very nice man as well as a wonderful scientist, and he smiles a lot anyway. So Mercludex B is a 20, I think it's a 20, it's got several dozen amino acids, again, the concept of those links in a chain, 47 amino acids to be exact, that are taken from the sequence of amino acids in the part of the hepatitis B coat protein called pre-S1. So it mimics the coat protein or the portion of the coat protein that hepatitis B uses to bind to cells at the NTCP receptor. And by creating this synthetic version of part of that protein, what he was able to do was block the receptor and make it inaccessible to binding by the whole virus. So it blocks the receptor functions of NTCP and therefore blocks entry of hepatitis B with or without hepatitis B. It is administered medicinally under the skin once a day. Next slide. And this is a picture also taken from Google Images, but has proper attribution with a very graphic depiction of what's going on. The Mercludex B is the red star blocking these bile acid receptors from binding the little green dots that are the bile acids, but also blocking hepatitis B from getting in. Now, the main point of this is to show that Mercludex B does result in a holdup of what's called the circulation, the enterohepatic circulation of bile acids that circulate from your liver where they're made down your bile ducts to your intestine, but then much of it is recirculated because of reabsorption in the lower intestine back to the liver. And so the red line in the graph on top shows that there is an increase, as you would expect, in the serum level or blood level of bile acids. You're holding up the ability of bile acids to get through into the liver cell again. And so we'll talk about the consequences of that for the safety profile, which actually turn out to be refreshingly minimal. Next slide. This is the earliest study of Mercludex that was published about 5 years ago in the Journal of Hepatology. And I'll just go very quickly to the first of the three graphs on the right side of the slide, looking at the patients who got what is the now approved dose of Mercludex B, 2 milligrams a day, showing about a 1.7 log reduction. Remember, log is 10-fold, so this is maybe very roughly a 50 or 60-fold reduction in the level of HDV RNA, a major reduction in the level of HDV RNA in the blood with 24 weeks of a very well-tolerated oral drug. In my little insert at the bottom, you can see that the viral RNA actually decreased by more than 10-fold in 6 of the 7 patients, that average of 1.7 logs, and actually became undetectable in 2 patients out of the 7 during treatment with Mercludex B alone. The results were a little better when you combine it with interferon and gave interferon alone, but here's an oral drug, very well tolerated, really having significant activity against the virus by itself. Next slide. And this is one of the 2 major studies of Mercludex B that's been completed now. This was a study looking at Mercludex B in 2, 5, or 10 milligram doses with tenofovir, one of the 2 mainstay oral drugs for hepatitis B, versus tenofovir with active treatment for 24 weeks versus and 24 more weeks of follow-up with tenofovir alone in all the treatment arms. On the left, you can see the median declines by log again in HDV RNA, the 2 milligram dose, very similar to the previous study, about 1 3/4 logs, somewhat more with the 10 milligram dose, combined with tenofovir. The tenofovir is controlling the hepatitis B. It's really not exerting any independent activity against the hepatitis D, as you can see from the tenofovir alone graph. And then the primary endpoint reached at the end of the study, 48 weeks now, with lasting effects from the previously taken Merclude-XB, either a 2 log reduction or undetectable HDV RNA at that last time point in the study. Quite robust with all of the doses of Merclude-XB here, higher admittedly with the 10 milligram dose. Next slide. And this was another study on Merclude-XB, looking at interferon now, the other one with tenofovir. And this became more complicated because of an extension arm that was added later. But look at the week, this is treatment for 48 weeks in all these arms, interferon alone on top, 2 or 5 milligrams of Merclude-X with interferon, and then Merclude-XB alone. And you can see that even the Merclude-XB alone at the bottom of the table on the lower left had almost a 3 log reduction in viral level at week 48. Remember, it was about 1.7 logs at week 24. So there's cumulative impact of Merclude-XB the longer you give it on viral load reduction. Of course, when you stop and you look again at week 72, there's still an effect, but it has been reduced. And on the right, you see the primary endpoints. I'll move on in the interest of time. And this is the extension study where they added 2 more arms, both involving 10 milligram daily bulevirtide, which became the new scientific name for Merclude-XB. And next slide. This gets a little complicated because now all the arms are shown. And you can see that for the 2 milligram bulevirtide, now called BLV, the primary endpoint at the end of follow-up with 48 weeks of treatment, 6 months of follow-up, was 6.7% for undetectable HDV RNA, but about a third still had at least a 2 log reduction. And the numbers were higher in the groups that got interferon, as you might expect. Next slide. I love this presentation from the AASLD recently. This is Merclude-XB for 2 years in the first 2 patients and 1 year in a patient who apparently could have been continued, but stopped for various reasons and had recurrent viremia shown on the left. What I love about this is these were compensated cirrhotics who have a real risk of decompensating and having their livers fail. And on the right, the 2 right patients had ongoing complete suppression of virus with normalization of ALT and even evidence of clinical improvement despite the existence of cirrhosis, like resolution of esophageal varices, which are swollen veins in the esophagus that occur as a result of backup because of the scar tissue in the liver, exerting back pressure against the flow of blood into the liver with a traffic jam in all of the other veins in that part of the system. So I love this, even though it's sort of a small case series, because I think it may be a guide to how we should be using Merclude-XB in the hepatitis B population, namely as a long-term maintenance therapy. It's targeting a host factor, which means that it would be hard for the virus to become resistant to this, much harder than a direct-acting antiviral, for example, like the ones we use in hepatitis C, where we have to use antivirals together to prevent resistance from emerging. And I think this is very tantalizing. Next slide. I have to go really fast now because we're running out of time. This was the eagerly awaited announcement of the European Commission approving Merclude-XB. It actually happened in late August, even though this announcement from the Hep B Foundation came on September 2nd. Next slide. And it is approved at that dose of 2 milligrams subcutaneously once a day. It doesn't really stipulate, to my knowledge, exactly how clinicians should use it, which is why I mentioned how I might choose to use it if it could be made available here and a clinician were free to use it for as long a period of time as you felt was in the patient's best interest. Another drug is lonafarnib, which is that prenylation inhibitor. I don't have time anymore, but I put in a little bit if you get these slides so that you understand what prenylation is, but it has to do with the addition of that lipid moiety to the large delta antigen protein. Next slide. This does have antiviral activity, as shown here in an early phase 2 study of the drug given with ritonavir to boost its blood levels, something that ritonavir does for many drugs, much more so with interferon, but activity even without it. The ritonavir is just boosting the level of the drug. It's not an antiviral against hepatitis B. And you can see nice composite endpoint data with either a 2-log decline in viral level and ALT normalization in fully 63% of the patients who got all 3 drugs after 24 weeks. Next slide. So that led to a study with Lambda Interferon, which was initially studied by itself. Next slide. And does have activity. This is allegedly, potentially, a more tolerated form of interferon. So it is still of some interest, particularly for this infection, with a 1 in 3 approximately rate of durable virologic response in this small study. The point being, next slide, that this led to a study of PEG interferon lambda, lonafarnib, and ritonavir for 24 weeks, presented very recently at the aforementioned Liver Association meetings. And there were 26 patients, 22 got to the end of treatment, as you can see at the bottom of the graph on the lower left. And at the end of the study, 23%, 5 of the 22, maintained an undetectable or below the level of quantification HDV RNA levels with very nice levels of success and viral suppression while on treatment. 77% achieved at least a 2-log reduction after the 24 weeks of therapy. And I'll leave you with the additional information on this slide. But this has led, next slide, to the next slide, to the formulation of an ongoing phase 3 trial, now going back to PEG interferon alpha, either alone, going from the bottom up to the blue color, all 3 drugs, and then back to lonafarnib and ritonavir alone for 48 weeks with 24 weeks follow-up. This study is currently ongoing. Next slide. And finally, a drug called REP-2139. Next slide. You can just show all those together, which is said to inhibit the release of hepatitis B surface antigen from the cell, and therefore potentially anything that is wrapped up within surface antigen, like a hepatitis D viral particle. Next slide. And there have been some very dramatic results with this agent reported in both hepatitis B and D over the last several years. Here you see very powerful suppression with REP-2139 given initially for several months, followed by PEG interferon, with universal clearance. You don't see that phrase too often, of HDV RNA during therapy, some relapses later on. Next slide. And next, you can just show it all at once. With hepatitis B surface antigen clearance prior to interferon just from this drug alone. Next slide. And we can show the whole thing. And again, this is the much quoted hepatitis B study, series of presentations over the years, staggered administration with upper left tenofovir alone versus followed by tenofovir plus interferon, followed by the 3 drugs. A NAP is a nucleic acid polymer, which is what 2139 is. It's a few nucleotides linked together versus all 3 together at an earlier time point. And the green lines will show this incredible reduction of HBsAg to very, very low levels or even undetectability. And the bottom line shows in green the increase in surface antibody as the surface antigen clears. Very provocative, of great ongoing interest, and with great potency against hepatitis B as well. Next slide. Finally, a recent presentation in a paper showing long-term functional cure of hepatitis B and D with a course of REP-2139 derived from the patients in those studies. Next slide. And I'll just let you look at this for a minute because I realize I've gone over, and this all summarizes the points that I've made. Toward the bottom, I'll remind you that treatment with interferon alone is available on a widespread basis. It can result in SVR in 25%, but you can never stop following these patients because late relapses can occur. We'd love to get Merclude XB approved in the US. I'm currently applying for a compassionate use basis with the FDA to use, working with Mir Pharmaceuticals to get this drug for one of my patients with advanced fibrosis who's very anxious to be treated. And as I said before, maintenance therapy may be needed. Lonafarnib is in phase 3, and the results with NAPs intrigue everybody and warrant further study. Thank you very much.
Great. Thank you so very much, Dr. Jacobson. We really appreciate your time and insights, and the presentation was very comprehensive, and we look forward to these future treatments as well. I know we're approaching the top of the hour, but if everyone can hang on, if you're able to, for just a few more minutes, we'd like to share Rachel's story. She lives in Texas and is living with hepatitis B and Delta, and she's graciously agreed to share her story with us. So we'll listen to Rachel and then have time for a couple of questions.
My name is Rachel Scott. I'm 28 years old and I live in Amarillo, Texas. I was born in Romania. I was adopted to the United States when I was 10 years old in 2002. In Romania, I was infected with hepatitis B and D. I remember being put in a line with other children and the nurses were using needles with other children, never changing even the syringe. It is felt that this is how I contracted hepatitis. I was diagnosed with hepatitis Delta only in 2019. My GI doctor has me come for a follow-up every 6 months for a sonogram of my liver and have my liver enzymes checked as well. My liver is healthy right now and the liver biopsy showed only rare fibrosis. I have multiple health and digestive issues for the past 12 years.
Sorry. I'm sorry.
That physicians are not able to connect to any cause. Because I am the only patient in my city with Hep Delta, I have not found anyone who can advise me about these unknown symptoms and conditions. The closest known doctor is 6 hours away in the Dallas area. Being diagnosed with hepatitis Delta was a huge shock for me and my family. I was aware of hepatitis B, but I didn't know anything about this kind of hepatitis. Because I have hepatitis B and D, I have to watch what I eat and am careful of chemicals and medications I put in my body and on my skin. Everything I eat is organic and non-processed. I am unable to eat anything that is not freshly prepared because my digestive system is so very sensitive. I work for an adoption agency as well as my mother, and she has become an advocate for testing for hepatitis D in the international adoptive community. She educates those who know they adopted a child that is Hep B positive or to those adopted that are positive and encourages them to test. We feel there are many like me walking around not knowing. I'm excited about the potential treatments in the future that if my disease begins to decline, that I might be able to take advantage of. Every day I pray for a cure for myself and others before it's too late. Thank you so much for giving me the opportunity to speak and to tell my story.
Thank you so much, Rachel, for sharing your story with us. We really appreciate your perspective and hearing the story of someone living with hepatitis Delta. And now we will take some questions for those of you who can stay. So Dr. Jacobson, we have a question. Which one of these drugs do you think is the best treatment?
Can you hear me all right?
I can hear you, yes.
You know, to really answer that with scientific rigor, we'd have to do comparative trials. We'd all like to get away from interferon in this field because of the side effects that it causes, but it remains the only widely available active drug. If you're asking me to imagine in the future what could be the best drug, it's really hard to do that. You know, these NAPs or nucleic acid polymers have been associated in the one series of studies that we have available with very high rates of clearance of not only hepatitis B, but hepatitis D. So it is very intriguing. Those studies need to be expanded upon and confirmed, but very provocative, as I said. I think Mercludex B looks very nice, particularly if one embraces the idea of a maintenance drug. I didn't mention, but there really are no major or even minor side effects with Mercludex B. Since we think that bile acids make you itch in people with liver diseases that result in inability to get rid of bile acids from your body because they get stuck in the liver or the bile ducts, we had worried that Mercludex B, by raising the bile acid levels like I showed you in that picture, might make people itch. But interestingly, they don't, which makes us go back and question, as we have for years in a very controversial series of articles that have been written, whether itching really is just from bile acid elevations, because they don't happen with Mercludex B, which is great. So again, I think right now, if I had Mercludex B at my disposal here in the US, which we don't yet, I think I would give it to all my patients with hepatitis B. And what I might do is give them an initial course of interferon with it to kind of jumpstart the whole thing. But if I knew I had that drug available for long-term use, given the admittedly anecdotal patients with cirrhosis that I showed, I'd be very attracted to the idea of continuing to use it. Lonafarnib is definitely an active drug. It does have more of a side effect profile, particularly GI side effects. And about 19% of the patients in that trial, which I didn't get a chance to show because I was out of time, did have to stop because of side effects, though not the GI side effects, interestingly. They had elevated bilirubin levels, which may have been from the ritonavir. So I can't really say, I think it would be wrong for me to say there is a best drug, but I can certainly share with you what I'm attracted to right now.
Excellent. Let's hope for continued approval throughout different countries of Hepcludex. Another question is are the once daily injections of Mercludex B, is that for indefinite, or is there a time frame on that?
Well, I looked in the approval language, and Tim may be able to help me here, but I looked in the approval language for a stipulated period of time for which it's approved. I know that in my application for use in my one patient, they're telling me that they can give me 6 months. But again, if given my druthers, I think the drug really, to achieve its optimal effects, needs to be used for a longer period of time. And as I said before, indefinitely perhaps. I think we need a lot more studies on what happens after you use it for a year or even 2 or 3 years, and then stop it. Perhaps we would get more durable responses, although we do get a few even now after 48 weeks of use. So I see it as a long-term use drug right now, unless we come up with something to give with it that knocks out the virus for good with the help of the drug given for a finite period of time.
Okay, excellent. And then it looks like we have one more question. Would you share your insights on why Mercludex or bulevirtide seems to only affect the virologic response of HDV, but not HBV? Should it not also block the entry of HBV as the NTCP inhibitor?
Well, that's a fantastic question. And the person's right on the button, so to speak, in terms of the question. So absolutely, there should be effects on hepatitis B. And indeed, those studies have also been done. There were some hepatitis B data embedded in the hepatitis D study, but I already went too far over to spend more time showing that, so I couldn't. The effects in the hepatitis B studies on surface antigen are actually quite modest, but that's not a deficiency of the drug. It has to do with the hepatitis B life cycle and the fact that our best drugs against hepatitis B don't really do much against the hepatitis B surface antigen, the clearance of which is, as I said before, the holy grail of treatment. So if you look at things like hepatitis B replication, you can see an effect, but it's that deeply embedded inside the liver cell hepatitis B surface antigen that can continue to be expressed from embedded sequences of the viral genetic material, even after hepatitis B replication has come to a halt. That's very difficult to get at. And so I don't know that Mercludex is going to be to have the special role in hepatitis B therapy that I think it should have for hepatitis D. We and many others are working very hard on developing new treatments that are designed to get to clear hepatitis B surface antigen. But Mercludex by itself doesn't really do that. But it does inhibit entry of hepatitis B into the cells, yes.
Okay, excellent. All right, I think that is all that was all the questions and all we have time for. So thank you so much again, Dr. Jacobson, for your presentation and your insights. Thank you so much, everyone, for attending. Just a reminder, a recording of this webinar will be emailed to everyone. And if you could, please complete the surveys that are in the chat box to let us know if you'd like to be added to our newsletter list and would like to be updated on clinical trial information, as well as the evaluation form for this webinar. And if you'd like any more information about hepatitis delta going forward, please be sure to visit our website or send an email to connect@hepdconnect.org listed here. So thank you so much again, everyone, for joining, and have a wonderful rest of the day.
Thank you.
Thank you.